Okay, perfect. So let's go ahead and get started. So thank you everyone for joining us for this week's AME seminar. Today we have Vijay Rajagopal, who is going to be talking about biological microscopy image segmentation. He leads the cell systems and mechanobiology lab at the University of Melbourne. And so we're in for a pretty neat talk on AI. So take it away, Vijay. Thanks for being here.
Thank you so much, Jorge. Really appreciate your invite. And also, I should really thank Zoom because it's allowing me to be in Texas for an hour before I head back to being in Melbourne, Australia. Right. My name is Vijay, and today I'm going to talk to you a little bit about how we harness AI for some of our work. What does hopefully my mouse is working. There we go. What do we do? My research group, the Cell Systems Mechanobiology group, we like to create digital twin models of cells and organs for basic science and clinical applications. A lot of what we do is focused on the heart. So we have computational models of cardiac systems biology, all the way from elementary calcium release events called calcium sparks, all the way up to whole heart modeling. And what we're interested in doing is understanding how the heart adapts as you go from birth into adulthood, old age, and disease. And so we have a bunch of projects you can see at the bottom here. We've been working on how the human heart ages, how the heart grows, what are some signaling processes that govern heart growth. And we've just started a project on developing heart with a colleague at Monash, but we won't have time to go through all of that. But just to give you a flavor of a sense of what the broad scheme of research is, I wanted to lay this out. In as far as we are studying how the heart works using using data analysis techniques that we develop. We want to develop tools and mathematical models that can be applied across different cell types. So uh, where there's opportunities to translate what we do into another application, we get right into it. So on the right here, we've worked on studying how cells form adhesions. We've also looked at cell migration, and I'll briefly touch upon that today. And of course, our work that we are doing in AI or the application of AI, we're using it in a bunch of different projects. I won't be able to go through all of them, but yeah, so that's basically the research group. It's developing computational models and computational methods that are for which we have some specific application in the heart, but also finding applications in other places. I want to just I should before I get into the talk for the day, I will give a brief introduction to my career. I did my PhD between 2003 and 2007 in the Auckland Bioengineering Institute, which is renowned for creating digital twin models of tissues and organs. My focus was on studying breast tissue mechanics. So I developed methods to create patient-specific models that you could then embed clinical breast cancer images in, and you can then track tumor suspicious lesions from an X-ray mammogram onto a 3D MRI image stack using these biomechanical models. Towards the end of my PhD, I discovered a book in the Institute library, which was about cell biology, and I decided I'm just going to massively pivot, and I decided to go and study cell biology. So I started off my postdoc in and really went for it. I was successful in getting an early career um, research award to develop a 3D model of a muscle cell towards studying diabetes-induced heart disease. I also spent some time in this place called Singapore MIT Alliance. It's with Professor Roger Kam at MIT, with a lab in Singapore where I studied microfluidics and looking at cancer cell migration. So after my stint between 2007 and I should say 2014 rather than 2012, I then was lucky enough to then get an academic position at the University of Melbourne, where I've been since 2014, and just building on all the skills I learned in my PhD and postdoc, and driving a research project since then over here. If it, I won't spend too much time on this slide, but if people want to ask me some more questions about career and career management, I can certainly give you some of my experiences. So, but to summarize what some of the things that I've learned about what it is to manage a career. You could kind of base this may not be novel, but it's interesting to be experiencing all of it, having moved through different countries and different labs and also different research interests. Um, what makes up a great research career is at the foundations, training at the best places to become technically excellent. You want to be building up a vision of what you want your career to be, what you want your research to be about. And it doesn't have to be clear at the start, but it will become clear as you experience science. And as you build up your research experience, develop a good, excellent team of researchers, not only as collaborators, but also if you're fortunate enough to create a research lab of your own, go through the process of recruiting the best people that you can who will really enhance your experiences. And all of that in a great environment, which sometimes you have to create. And I certainly think the stuff that you're doing, Jorge, with this seminar series is one of those examples where a research environment is excellent environment is a place which allows people to share ideas, stimulates new ideas, and enables people to think bigger than their individual kind of next deadline. So yeah, certainly we can talk about all of that in the questions section. I like to put my acknowledgement slide before the talk because I tend to just completely forget about it by the end of the talk. So a lot of what I'm going to present today is about work done by Afshan Khadangi, who did his PhD with me. He's now in industry here in Melbourne. You can see the papers I've listed here. They're the papers that he published in this area. He was co-supervised by me and Thomas Boudier, I think he's in France now. And towards the end, I'll be talking a little bit of the stuff that's unpublished, but it's by my PhD student Aidan, who's currently working with me. All right. So what am I going to talk about now that I've talked a little bit about my career and acknowledged what work I'm going to talk about? I'm going to walk through in this presentation what I think is really exciting about research in bioengineering. I think people should just stop working on whole organs, and everyone should be thinking about the single cell because it's really an age of big data, and I'll explain what I mean by that in a minute. And then in that, I'm going to talk in that challenge, in that sort of excitement of big data comes a lot of microscopy images. And I'm going to talk to you about what kinds of images you can collect and what challenges they bring and why we need AI for that. I'll be then just briefly giving you an overview of the couple of places where we've innovated with AI or to advance our interests, which is to build a virtual model of a cell. And then towards the end, it'll just be these very one slide snapshots of the wider research that we're doing. Partly it's to let you know that we don't just do AI, but also it's a good plug for my students because there's a lot of great stuff happening and there's just not enough time in one talk to talk about everyone. So let's just do a quick sneak peek kind of session at the end. All right. So we're in 2025, and the big data explosion is happening in cell biology for the last 5, if not 10 years. And it's really exciting because what you can do now is you can visualize or measure cell biology down to single molecules and all the way up to whole organ. On the left here is a electron microscope image stack of muscle cells, and each cell is one of these very curly shaped objects. But inside it, what you're seeing are mitochondria and myofibrils that make up the muscle cell. There's even more detail that is just not I'm not able to present to you in this GIF image. On the middle, you have these techniques called what they call multi-omics, which is basically using mass spectrometry to measure the abundance of different proteins, metabolites, transcripts inside cells of tissues. So you can get big data on all the components that make up a cell. And the third thing is we also have this capacity now to image cells at high resolution, if this movie will play. Down here, what you're seeing is this the leading edge of a cell, migrating cell, and you can see this membrane here. The edge is basically membrane ruffling, the cell membrane ruffling, and the inside you have molecular machinery that drives that ruffling and helps the cell move forward. This is just one small aspect of the types of measurements you can do. You can measure in pH of individual organelles now. You can measure not only how the cell moves, you can measure how the cell divides. You don't only get to see how the cell divides, you can also see how the whole embryo or how the whole organ grows on an experimental platform. And you can image these things for hours. So you can get lots of data. And the problem is the excitement is that you have lots of data, but the problem is that we need to be able to analyze them if we're going to make some discoveries. And the first one is just being able to process the data using high throughput methods. And the second challenge is, can we connect these different data sets? Because in the end, what all clinicians, basic scientists, basic cell biologists, and physiologists want to do is we want to understand the physiological state of an organ or a cell so that we can then make predictions or develop treatments that can get us back to a healthy state if we're in a disease state. And so understanding data sets is one phase of one aspect of science. Being able to connect the dots between those data sets to understand physiology is that next level challenge. And bioinformatics and statistical techniques have been so far the method of choice because of the sheer size of the data and the complexity of biology. But there's this increasing push towards an interest towards creating a model, virtual cell model, because if you can connect these data sets mechanistically, using these virtual cell models, you can then start to make inferences that are mechanistic. You can start to say, hey, if this particular process is going wrong, this is a mechanistic drug target that will not only alleviate symptoms, it will also make sure that that mechanism is not going awry. And so while there's a still robust need for statistical methods, there's a need for statistical methods to link with mechanistic methods and mechanistic models. And so there are people all around the world, not just in Australia, who are working towards these virtual cell models, and we're one of those people as well. And what we've started with is wanting to build a virtual cell model of a single beating muscle cell, because cardiac muscle cells, they may seem like they're just cylindrical shaped cells that just contract every heartbeat, but there's a lot of dynamics in it that helps maintain the heartbeat. And if the muscle cell is not working well, then it leads to cardiovascular complications, which then lead to eventually lead to fatalities, which we're all familiar with in our families and and friends. So with this interest in building this virtual cell model, we've been working on it for the last 10, 15 years, starting from my postdoc years. We've got some stuff done in calcium dynamics. We've been looking at energetics and metabolism, and we're also now looking at the organization of muscle cell myofibrils and how they impact on cell mechanics as well. And when we're building these models, one of the fundamental steps you do is you need to create the model, the geometric model of the cell. And I didn't realize this when I proposed this project as my early career grant, that would actually end up becoming a grand challenge, just to create this model of a muscle cell or even any living cell. And what is the grand challenge? Well, I'm going to explain. There are quite a number of challenges, but I'm just going to bring to light three challenges. First is that the cell is a crowded space. Here is a 2D image. It's from an electron microscope imaging system. And what you're seeing is a crowded organization of these striped patterns called myofibrils. They're the proteins that contract every heartbeat. And then these lozenges, I call them dark gray lozenges, they're in mitochondria. They produce the energy for every heartbeat. What you may not be, for the untrained eye, I may have only pointed out two, but there are actually several components here. If I zoom in a little bit more and started to label all the components that make up the cell, you start to realize that there's more than meets the eye. It takes a while to realize that it's just packed with content, and every bit of content in the cell has an important function. Here you're seeing myofibrils I've already mentioned. The Z discs are proteins that are basically anchoring proteins for the muscle cell to contract against. Mitochondria I've already mentioned. Lipid droplets produce energy, also are sort of energy sources. I haven't highlighted these things here. This is your dyad, which is the localized microdomain in the muscle cell that releases calcium for every heartbeat. So there are lots of different components, and they're really juxtaposed against each other, making it quite a challenge for image segmentation algorithms to resolve. The second challenge is that you don't actually get all of the data in one imaging modality. So here I have two sets of images. One is electron microscopy. Another is I'm showing you a confocal, but there's more current ones like super resolution that we can talk about. And electron microscopy generally gives smaller volumes field of view and low contrast, but it has everything in it. But it's so hard to see through all of it because of the crowded nature of the cell that you don't really know what sometimes some things are. On the right-hand side, confocal or super resolution, it's at a larger field of view, great contrast, but the antibodies that you use to stain and image things means that you can only stain and identify, say, 3, 4, maybe 5 different components. I mean, and I'm stretching it for 5. And so you have this challenge that you'll be able to do some imaging on some tissue with EM, but you can't really reuse it for super resolution or confocal. So you can't get all of the detail in one go. So that's another challenge. And bear in mind that since you can't reuse the tissue for EM on super resolution, you will have to use another tissue sample. So it's not even the same cell anymore that you're looking at. The third kind is that third kind of challenge that I'll talk about today is that cell structure is not static. It moves, it changes, it remodels. Here, what you're seeing is a data set collected from my colleague Senthil Arumugam here at Monash. He's a group lead here who does really advanced microscopy. And what you're seeing are these green lozenges, which are mitochondria moving around as cells are migrating on a 2D surface. And if you're going to be able to study the cell for in its full physiological sort of characteristics, you have to capture cellular structure dynamics as much as just where it is in any given moment in time. So what I'm going to do now is I'm just going to go through um, some of the work we've done using AI in addressing these challenges. So I'll start with the problem of cells being crowded, and I want to just do a brief sort of uh, um, overview of what we're talking about, just to give you some context and some the significance of what we're doing. What I'm going to talk about is segmenting electron microscopy images of muscle cells. So these are heart muscle cells. This is a typical example of what a heart muscle cell looks like, at least on a 2D image. And what they do, heart muscle cells, well, if for those who are not familiar with the details of how the cellular details of the heart, basically, if you have your whole heart and you started to look at one of the walls of the heart and you looked into the mid-wall region, that mid-wall region contains cardiomyocytes or heart muscle cells. And those are the muscle cells that beat every heartbeat for your heart to contract. Their arrangement inside the heart governs how the heart pumps as a whole, and their arrangement also affects how they themselves are internally organized, which is something that has become sort of common knowledge thanks to this field of mechanobiology that's been going on for the last I'd say 20 years. So we're going to talk about muscle cells, and this is the typical image of what a muscle cell looks like. You've got your cell nucleus, you've got your T tubules. They're the invaginations of the cell membrane that activate the muscle cells contraction, excitation, contraction, coupling. And you've got mitochondria to produce energy, and then your myofibrils. When we started working in the space, we set ourselves the challenge of segmenting the Z discs. I've already mentioned what Z discs are before, but just a reminder, they're basically these stripes that you see, black stripes that you see in our EM images, and they're basically anchoring points for the proteins of the myofibrils, so that the muscle cell can contract against them during each heartbeat. And any misarrangement to those Z discs can then cause malfunction and contraction, which then affects how your heart beats. Now, the thing about these Z discs is they're everywhere, but they're also very thin. So in terms of the serial block face imaging data sets, which is a particular volume EM data set, electron microscopy data set, hopefully you're able to see the stripy bits here, right? They're everywhere, but then they're in amongst all these other details that are also of similar intensity and similar texture that one has to really work hard to segment all those structures out. A little sort of fast forwarding here so that we have time to get into the AI. We spent about 2 years developing image processing algorithms. This is back in 2015, maybe 16. We did some image processing based methods to segment these Z disks. It might even be 17 actually. And Afshin, who did all this work, really worked hard to develop an algorithm that would track these Z disks, and the what you see on the right is an output from his work. But what we found is that you really, when you work with algorithmic methods, it's really only sort of translates to another image stack that looks just like your image stack. And so it's a lot of work for not much scale up. We also worked through some machine learning methods. So there were there continue to be these freely available, really powerful machine learning software packages that you can use. And what we tried is to manually annotate some of our images, and then we would um, test how machine learning algorithms perform as you increase the number of ground truth images. And you can see that it is possible as you increase the ground truth images to improve your metrics on performance in segmentation. But the challenge with it was that we had to know what combination of features would work to segment these data sets. And again, that also is not great for scalability, because once you have a different data set, you'll have to think about other features to extract. So this is why this was sort of a part of our learning process of why and how we should use AI. We didn't want to just jump in and use AI for the buzzword. We really wanted to understand whether it's going to help us or not. And so at the time that we'd finished working on this image processing and the machine learning algorithms, we knew that UNets were worth a shot. And of course, ImageNet is a competition where people have been trying basically trying different AI models to see if they can segment all these structures of cells and of images of cats and everything else actually using AI. What we wanted to do is see if we can segment our own data sets using UNets. We knew that it was data hungry. I don't know if it was at the same time that someone had already published, or I knew that they were going to publish a UNet-based segmentation of electron microscopy data. They had done it for HeLa cells, which is another cell type that's used for in experimental biology labs. And they had used they needed to hire so many summer interns to do manual annotations to get these things to work. And also they needed like 10, 15 AI models for different parts of the cell to segment out that they needed to train. And it was this very unique data set. It's this really big, unique data set. And my take on that was that if we're going to build, not every lab is going to be able to do what they were able to do, hire as many summer interns as they did, have access to the type of data that they did. So we wanted to really figure out what can we do with UNets that would help us with our work. And we don't have many people who can do manual annotations. So one thing we wanted to do is figure out, can you train a UNet with fewer data sets? So Afshin went away and tried a bunch of things, and he came back with a solution. If you have a look at a UNet, between each node of that UNet, you have these ReLU units, right? These I always get my acronyms wrong. It's rectilinear rectifiable linear units, or yeah, I think that's what it is. So what he realized is that instead of just keeping that as a linear, hard-coded linear function, piecewise linear function, why don't we just turn that into a trainable function? So we now call this a trainable linear unit, and that brings about two parameters. And what we found is by just opening up that parameter space, we're able to now train models with fewer data sets. And here's performance metrics, just accuracy and F1 score with our technique. We call this EMNet, but really, to be very transparent, it's not that we segment EM data, that there's nothing really novel about this UNet being used for EM data. It's really the trainable learning unit, that trainable linear unit that we can that makes this a powerful algorithm. So if you wanted to use it for your own images, then have a look at our work and see if you can implement the algorithm. It's all available on GitHub as well, by the way. So here's EMNet's performance for accuracy and F1 score. Its different variants are shown here at the bottom. And then we compared it against UNet, VGG, SegNet, and ResNet. And you can see that EMNet does indeed perform quite well. It typically has as you go through the number of epochs, you can typically see that EMNet does learn a bit faster than the standard UNet model and it's like and VGG and so on and so forth. Now, of course, when we did this and we submitted this this work into the one of the I think it was the IEEE journals, we were aware that there were other models being published saying that they had developed the best model for electron microscopy image segmentation. And so much so that people who saw us give talks about us talk about wanting to develop AI models for our EM segmentation, they would say, "Why are you doing one? There's all these other ones that are already existing. I don't know why you don't waste your time on it." So that really made us think about, what should we really, can we just pick one that's going to work? And we found that there really wasn't any benchmarking done for the performance of AI models in segmenting electron microscopy images. So what we decided to do was do that work. So EM Stellar is our attempt at benchmarking AI models for electron microscopy image segmentation. And what we did is set this up as a Google Colab, where you can pass in a raw data set with some what the EM Stellar does is it then applies different trained AI models that are on that we've implemented on the system, and it will tell you the performance of each model to try and choose the best one. So this process of testing out different models and then outputting the result, it really helped us learn what makes for a good model. Interestingly, you'll see the answer is actually quite complicated. So just to briefly summarize what types of models exist in in EM Stellar, we put together this combination of VGG, ResNet, UNet, CDEEP3M, and of course, CDEEP3M was another new one that had come around the same time as EMNet. So we added all of these models together, and we had these two publicly available data sets. This is the cardiac one that was published by a colleague at NIH, and this is another electron microscopy data set that's been used as an EM image segmentation challenge data set before, which is from brain tissue. And so we took these two different data sets, and then we basically studied the performance of our different implemented models across a bunch of metrics. Rather than just doing accuracy and F1 score alone, we said, well, let's just examine as many metrics as we can. And so on the left-hand side, you can see Jacquard index, NPV, sensitivity, the VRAND score, F1 score, and the heat map tells you how good any one model performs. If you look at the CDEEP3M, it's mostly in blue, but it performs well with sensitivity. If you look at EMNet, it performs well, but it's really all across the board kind of complicated. It basically shows to you that there isn't one clear model that you can say is the best model. You can try any of these models and you get variable results. So this one on the left is for the cardiac, and the right is for the nerve cell, the neuron. And you can see again, even across when you go across data sets, the performance changes. So for CD3M didn't work so well for the muscle cell, but here it seems to do a better job with the neuronal data set. We also looked at convergence times of these models, and you can see the blue ones are the cardiac ones, and the pink ones are your neuronal tissue ones. And right away, you can see that the convergence times are certainly smaller for the cardiac data set than the neuronal data set. So there's something about the uh, image properties of the cardiac that makes it faster convergence time than the neuronal one. One could argue that the cardiac structure is quite repetitive when you compare it to neuronal data sets. And maybe it's that the structure of the cell itself makes for the difference. We then asked, well, what about the size of the layers, the size of the model? How does that affect the performance? Here again, blue is or cyan is cardiac, and pink is neuronal segmentations, and the diameter of the circles represents the number of parameters. And you can see if you look at CD3M, it's all the way over to the right here and at the top. So the number of operations increases in these models, but the performance is, comparable. And then if you look at towards the right, the performance is quite high for EMNet. It has much fewer parameters than CD3M. So what this basically says is having more nodes, having more parameters doesn't necessarily mean you'll get a better fit. I think that's something that we all do understand about overfitting. And what we've shown is by choosing to just make the ReLU into a TLU, you can actually get a lot of performance out of it without blowing up the number of parameters. Of course, those are all metrics, but what you want to know is did it really work on your data set? So here I'm showing you the performance of in actual segmentation results of the different models. Where there's red dots, that's a false negative. Where there's blue, it's a false positive. So you can see that for each raw data set, you do, even with EMNet, you will produce false positives and false negatives. But you can see that there are some models that produce more of them than others. Right? Let me come back. Here we go. Here's a look at the CD3M compared to EMNet for a cardiac data set. And what you can see is in CD3M, we find for cardiac that it produces lesser false negatives, but EMNet produces more. So the story is not that one is better than the other. It's that you get different behaviors with these AI models for different types of data. And so the conclusion that we came to is you should really just the analysis that we did really showcased why you want something like EM Stellar, because it allows you to then automatically just run a bunch of models and see what's going to work for your particular data set without getting too technical about it. And one thing we found that works best is actually to take all of the results from the different models, and then you do average voting or maximum voting, and basically what we call ensemble, taking the ensemble of models, which is this leftmost result here, you find that you just reduce the number of false positives and false negatives by just doing an ensemble of models. How am I going for time? You're okay? Going well?
Yeah, about 10 minutes.
Okay, 10 minutes to go, is it? All right.
Mhm.
So I'm just going to smash through the last two challenges here, but I did really want to spend a decent amount of time on the first part. because I think it's important to understand what the behavior of AI models and to be systematic in how you work with them. I'm now just going to briefly summarize the other two challenges, like what we've worked on. So here, this is looking at Grand Challenge 2, Part 2. How do you integrate two different types of data uh, of a cell, and it's not even from the same cell? What we did here is we actually took statistical techniques um, to address this problem. Specifically, what we did is we had a look at all all of our confocal images, which contain the distribution of particular proteins that we're interested in that are hard to visualize in our EM electron microscopy data. What we did is we took those images and then we did extracted the proteins of interest, and then we did a analysis of the distribution of those proteins relative to organelles that we can observe in our electron microscopy images. So this is what we call spatial statistics. And by studying things like the nearest neighbor distance between different proteins in our confocal image and their relative distribution to other organelles in the cell, like z-disks, we can then create a statistical model of the spatial organization of the cell. And now that what that allows you to do as well is you can basically tackle the problem that you may not get the images all on the same tissue block. So by collecting 5, 10 samples of these images and extracting the statistics, you can now put a statistical distribution of the organelles that are in the confocal. You can simulate them onto your electron microscopy. So we developed a workflow for this, and starting with your raw confocal images, extract your particular proteins of interest, and then calculate their position relative to other organelles that you will find in your EM. And then you simulate those proteins on your EM data set, which you've also processed in a similar fashion. And what you come to is something like this. So here is our electron microscope image. And I'm zooming in here. These red dots are simulated ranitidine receptors that represent the statistical distribution that we found in our confocal. So we now have an ability to computationally integrate different imaging modalities into one so that you can build these 3D models. Finally, Grand Challenge 3, which is this movie, here's what we wanted to do was to develop algorithms to segment, or develop a way to segment and track these mitochondria. This is what the real image, a close-up, looks like. Really exciting data here of the movement of mitochondria as the cells migrate. That's a lot. There's a bit of color that's added to this using a color table, lookup table. This is what the real image looks like. Low contrast, blurry, noise. How do you segment these data sets? How do you segment out manually where a mitochondrion starts and where it finishes? These are all challenges that we were facing. And we ended up, and again, I'm just going to run through it as a summary. We ended up trying a bunch of algorithmic methods that are out there and realizing that they didn't really work for our data because of the low contrast and noise. And so what we did is we developed a method to create fake images that look like our real images. And we're calling them mitomimics. And what that allows us to do now is to generate a whole lot of ground truth data without any manual annotation so that we can then segment our data sets. So on the left is a raw data set, middle is our data set, and the right, our segmentation, sorry, after training an AI model using micromimics. And on the right is segmentation using current state of the art. And so what we can do now is segment each mitochondrion. We're able to track them. We're able to determine where they fizz, where they separate, where they join, with no manual annotation whatsoever. So I'm going to summarize uh, what I've talked about today. Um, we've talked about, I've given you hopefully a exciting introduction to why you need to move away from organs and start studying cells using bioengineering. There are many relevant and exciting, AI relevant and exciting challenges that are presenting themselves in this big data explosion in cell biology. I've just given you 3 examples of how we've tackled them: EMNet, EMStellar, and micromimics. We are leveraging these things now, these techniques now, to build our understanding of how the cell works. We're looking at calcium signaling in muscle cells so that we can understand how calcium itself drives gene expression. So the middle image here is an experimental data set or experimental measurement of calcium concentration in the cell nucleus, the one in red, and our one starts to recapitulate that. So you can see how our models are starting to reproduce reality. We are continuing to work on the muscle cell mechanics. This is a AI trained model segmenting all the zetas in our data set now. What we're also able to do is segment all the myofibrils now, and we're interested in understanding whether myofibril organization, which is not as parallel as we used to think, whether that actually introduces a twist to the muscle cell, not just a twist in the heart, but also down at the single cell level. And we're working on energy metabolism. We've been doing some modeling of energy metabolism, and we're coupling that energy metabolism to cell mechanics in this project where we're looking at cell migration. With that, I want to thank you for your time, for giving me your time, and I'm open for questions.
Great. Let's thank Vijay for very interesting, very diverse